Hello, everyone. Welcome. Thank you for joining us today.
Without further ado, I will introduce Mariana De Niz, who I am delighted to introduce today. She's born in Mexico City and completed her degree originally in immunology at the University of Glasgow, but the focus of her research really has taken a sort of tropical medicine and infectious diseases bent after she did a master's at the London School of Hygiene and Tropical Medicine. After that, she won a scholarship to the University of Bern through European Molecular Biology Organization, EMBO. They supported her PhD at Bern University in Switzerland, where she focused on applying a number of imaging techniques to further identifying the life cycle of Plasmodium parasite, which is the parasite responsible for malaria. So again, in keeping with that tropical diseases theme, intravital imaging, lots of optical projection tomography, photoacoustic imaging, just a wide array of imaging approaches Mariana is very familiar with. And she's received a number of awards for her PhD work, and then continued her career with multiple postdoc positions. So currently she is the manager of the Nikon Imaging Center for Advanced Microscopy at the Feinberg School of Medicine at Northwestern University. But in addition to her research, she is dedicated to democratizing microscopy, so increasing accessibility regardless of nation, gender, race, or disability. She has a focus on her efforts in scientific communications, including the acclaimed Latin America scientist interviews that she did for the Focal Plane Group, and also the recent publication in the Journal of Microscopy, which actually reflects the BINA meeting that we had last year in September in Mexico. So she's currently the chair of Outreach and Integration for Latin America Bioimaging, and also the chair of Diversity, Equity, and Inclusion for BINA, as well as being an active member of both the Communications Working Group and the Training and Education Working Group. So Mariana is a tour de force, and at this point I will hand it off to her to share her presentation. Thanks, Mariana.
Thank you so much, Nikki. That was a very kind introduction. I hope that everything goes smoothly. My computer is old, and so recently it's been complaining about too many things happening at the same time. So let's see. So yeah, today's talk, I mean, me and Ivy, whom I will introduce a little bit later, we wanted to, so some of you attended Michelle Digman's session last week, where she presented her work, and she's the leader of LFD, and they organize a very interesting course that we both attended, and we were both financed by BINA. So I'll mention a little bit, I mean, I'll start with, talking about, so this is more or less my outline. I'll tell you a little bit about my research, but just as an overview, and then I'll go to the relevance of the different aspects of fluorescence dynamics to study host-pathogen interaction. So I've been aiming to attend this course for a while, and so I'm very happy that I finally had the chance to. And then, of course, the main aim now in my current role. So there are still research questions that I want to address based on this work that I had been doing. So the course was very inspiring in that sense, but of course in my role now at Northwestern, we also support a lot of projects, so even things that I wasn't familiar with or don't necessarily work a lot with, so a lot of the things are subcellular, we still support that, so it was great to learn and be able to bring this back here and better support our users with the range of applications that they need. So some of you might have been at my talk, which I gave during Women's Day, but so a recap. So basically I did, as Nikki had mentioned, I did my PhD. So since always I've been super interested in infectious diseases. And so these are complex, so all my focus was in parasitology. So these are parasites, and they have complex life cycles, at least most of them do, but the ones I studied definitely do. Which means they undergo different stages of their life in different hosts, in different host cells. So for instance, malaria, it starts with the bite of an infected mosquito. The parasite travels to the vasculature from the skin and then finds its way to the liver. And then it produces this so from one single sporozoite that invades one hepatocyte, it starts hijacking organelles from the hepatocytes, and then it builds up. It's supposed to be the fastest replication rate of any eukaryote, from one parasite to 100,000 or so progeny in about 48 hours. And so once this is ready, so each of these, so all the progeny here that you see are, once they egress from these bubbles that are called merosomes, they each are able to invade one red blood cell. So obviously not only one sporozoite invades, but many. So you can imagine, so that's why quickly you get very sick once these parasites leave the liver, because they each go on to invade red blood cells, and then the red blood cells, they undergo different modifications. They stick to the vasculature of different tissues, and then they burst and release more progeny. So there's another round of asexual replication. This goes on forever and ever, and then people, yeah, I mean, depending on the species and the strain, you might survive or not, you might develop different symptoms or not, and so this is critical. So all of these involves a lot of interfaces, a lot of different lipids, proteins, organelles. So in terms of materials that are interacting with each other, this is super interesting biology. And then of course there's the stages that take place in the mosquito, right? So at some point this parasite decides to produce sexual forms, and then if you are infected and you are bitten by a mosquito, they take up these forms, and the sexual forms then replicate within the mosquito midgut and then produce another round of infection. So this mosquito is now infected and can go and bite another person, and then this person gets malaria, right? So that's more or less the general idea. In this context, the work I've done has ranged really a lot. So this is a hepatocyte, and there you can see one of the examples of one of the things I studied, which was hijacking of the Golgi. So the parasite forms its own, what it's called the PVM, the parasitophorus vacuole membrane, and it uses that to interact with the host organelle. So you see at this point that, say, all this Golgi shown in green is not equally distributed around the entire host cell, but is all sort of piled up close to where the parasitophorus vacuole membrane is. So that part of biology is super interesting. And so I had been aiming to do third harmonic generation to study that for a while. And then, of course, that's one of the topics that we studied at the course. Okay, so apart from that, as Nikki had said, especially when it comes to the blood stages of infection, there's a lot of interaction of these parasites with the vasculature. It induces a lot of tissue changes. But in this case, and I'll talk about a different parasite in a second, in this case, this particular parasite, most of its effects are on, well, they are through the vasculature, and then of course, damaging the vasculature causes issues with oxygenation, and it can cause other downstream effects on the tissue as a whole, on tissue homeostasis. But that's something we wanted to measure. And so far I had been measuring that with intravital microscopy based on fluorescence, right? Not based on any of the intrinsic properties of the tissue, but rather we have reporter mice and reporter parasites that we can see in this way. So with intravital microscopy, I was able to explore how the parasite interacts with the vasculature of the different organs. And we also had bioluminescent parasites to be able to see their distribution when they're able to sequester, like this little wild-type mouse, when they are not able to sequester, like these SBP knockouts, and then what happens when you complement or not. But basically these different patterns and what's the implication of the parasites sequestering at the vascular endothelium or not. So in that sense, the host pathogen interaction part and the intravital microscopy were the takeaways of them as tools to study these relationships. And then I moved on to study something else, which was the Trypanosoma brucei. And so this differs from malaria in that the Plasmodium parasite, except for the form that is injected by the mosquito, everything else that happens in the human or in the animals that it can infect is intracellular. It's intravascular and intracellular. In this case, with Trypanosoma brucei, it's not intracellular and it's not intravascular. It goes away from, so it's free living, it has a flagellum, it moves around, and it's able to cross the vascular endothelium quite easily. So one of the main things that it does, and this was one of the papers that we published more recently, is that it's in crossing the vascular endothelium, it changes. So this was done in the lab. The finding that the adipose tissue is a huge reservoir was a finding that my former boss, Luisa Figueredo, and her team did. And so my project was all about so I've always been super excited about this interaction with the vasculature, right? So the vessels are sort of highways for pathogens to move around the body. And so one of my questions was really trying to decipher how the parasite crosses the vascular endothelium, whether you can manipulate it by blocking or not different receptors. But definitely one thing that caught my attention was how heavily remodeled these tissues are once the parasite crosses. So for instance, in the adipose tissue, you get a huge reduction in the lipid droplets, in the size, in the form. It completely shrinks the tissue, right? So this is a whole new avenue of research in Luisa's lab. And from my point of view, what I became interested in was how the viscoelastic properties and the mechanical properties of the tissue and the parasite influence each other, right? So the parasite starts changing form. It becomes more enlarged, it becomes bigger, it changes the way it moves, and whether and how that is linked to the changes in the actual environment with which it has to interact, that's an interesting question that I'm trying to address with mathematical modeling at the moment. But again, referring back to the actual tissue, so the composition of the tissue, the properties of the tissue change. And so I thought, fluorescence dynamics would be a great way to study that. The other organ that changes dramatically is the pancreas. So that's another huge reservoir. And is, again, completely remodeled. So how, for example, its composition in glucose, in insulin, etc., its ability to interact with these changes too. So anyway, those were outstanding questions for me. And this is just some intravital images of the work I was doing. You can see here a little bit there, the collagen structure. So we were studying this with second harmonic generation already. And here you can see just flow of these parasites within the vasculature, right? So there's a lot of interesting questions. This I did in all these different labs. So both the malaria and the trypanosoma in the labs that you see here. So I wanted to acknowledge everyone there. And right now I am managing the Nikon Center. I work for Dina here. Many of you know her, and this is the team. Each person supports different things. And one of the things I support is multiphoton microscopy, light sheet, and so on. But FLIM is one of the things that I support too. So relevance of these to our method. So this is more or less, I took a screenshot of the program of what we covered in Michelle's fantastic course. And so this is more or less how I organized it here. So anyway, in terms of supporting researchers' needs, so this I already spoke about my own work in the context of host-pathogen interactions, but supporting researchers' needs at Northwestern. So these were all the different topics. So for example, fluorescence correlation spectroscopy, raster image correlation spectroscopy, fluorescence fluctuation spectroscopy, fluorescence lifetime, and phase or FLIM. So this was super helpful. I had been supporting already several people with FLIM, but I had difficulty understanding how a phaser worked and how to best provide that particular support. And the other thing I had been interested in, and actually this I first heard from Leonel Malacrida. So when I was at Pasteur in France, he visited and he was discussing what the diver microscope can do for deep tissue imaging. And I was very interested. And then when I saw that it was part of this program, yeah, I was part of that practical and I really enjoyed it. Yeah, combining lifetime and hyperspectral detection, bioluminescence phasers, this is also something that I think will be really relevant for people here at Northwestern. And then, of course, instrumentation for in vivo multiphoton microscopy. So this is just one of the examples. This is work that was done in the Graton lab of how they used deep imaging with to study FLIM and third harmonic generation. Basically, they were studying the effects of Western diet on liver tissue, and this is not based on fluorescence. This is metabolic imaging using FLIM. So that allows you to measure the ratio of free to protein-bound NADH, which result in different lifetimes. So you can study them using FLIM. And so that has been used for several different things in my, so based on the work that I described to you and what I wanted to study, for the pathogens, this was perfect for me for many different things, right? Especially the changes in the composition of the adipose tissue and pancreas. As I mentioned before, but of course, I mean, the applications now have expanded enormously, and it's relevant to what researchers here at Northwestern are doing as well, Alzheimer's, cancer, and so on. So it's a widely useful technique. And then the other thing was third harmonic generation. So what that allows you to do is really study better the interfaces. So it's also label-free methodologies, and so to study the interfaces between water and lipid, or water and protein, or lipid and protein, right? So that was more what I was thinking, and the support that we are asked for here is more at the cellular level. So anyway, that's third harmonic generation, which I hadn't had so much experience with. Second harmonic was fine, and we discussed it over there as well. And then the other was this practical. So there I attached a picture of our practical. I don't think Ivy was part of that group, but I was with some other friends of ours learning how to use Phaser. So that was great. And then it was great to see all the tools that the Graton team has developed overall, right? Not just in terms of software, but also, I mean, everything, right? In terms of hardware, in terms of probes, in terms of software. So Michelle and Enrico and their teams have done really a lot for the community. And so, yeah, that's what I was going. So I mixed them up, the order, but basically, yeah, this is what we support here at Northwestern. And so it was helpful to get an idea of everything that's happening. And so here I attached some pictures, so that I will now introduce Ivy, but I just wanted to mention, so these are different pictures of the actual event. So, I mean, it was a great event, not only because of the theory that one learns, but also the people that one meets. So I was able to meet Ivy, of course, and we've been in touch a couple of times. I think so Enrico's and Michelle's teams were very open to collaborating, to being contacted if we needed help, and I've contacted them a few times for help when there are users whose projects, I have doubts about how to best support. I attached here some pictures. So this is Belén Torrado. She's Uruguayan. She knows Leonel as well, and so we were able to chat a bit about these techniques. She was introducing the practical of the diver. So there I was able to ask all my questions of like, how do you do this deep tissue imaging for label free, right? I met also Julia, who I had, she was one of the main persons supporting the instrument here that we have at Northwestern. And so to meet one-to-one was great. And of course, meeting Enrico, I mean, it's different to read all the papers and then you get to meet this person in person and then chat with him. He was really, really nice. And then just as a coincidence, so Hong Tao here in the picture above, he was my predecessor in the position I'm currently in. And so he came from that lab and he happened to be there when I was there. So I was like, I've heard so much about you. So anyway, that was that. And then, well, as a thanks to, of course, the entire team from Michelle and Enrico, you can see them here, to all the participants who were really great. And here, this picture we took precisely for the BINA, to thank BINA for their support. So they made it possible that me and Ivy attend this course. And I think it makes a lot of difference. I think in terms of impact, I've seen already a difference so today, precisely in the morning, I was helping someone with the film work they were doing for material science, which is not something I often have done, material science, but the principles are similar. So that was very helpful. So I think it will continue to grow. And with that, I think that was the end of my part. And we have another 30 minutes for Ivy and for discussing a bit the impact of BINA and the impact of this particular course. And with this, thank you for your attention. I'll stop sharing. Great. So let me just, it got stuck. Okay. Perfect. So how do we want to do it? Do we want to make, so any questions, or we directly go to Ivy?
I think it's whichever you would like to do. The floor is yours at this point. So,
okay, maybe let's switch to Ivy. So let's move to Ivy so that she has enough time, and then we can couple all the questions together. So Ivy, if you want to start sharing. So, I mean, I was very happy to meet Ivy at the course. We didn't know that we were BINA awardees until later. I think we started talking and it was fun, and we started chatting about science. And I was already very impressed with all the work that she had done. So she's been, I thought I was versatile in my career, but then she started telling us like, oh, I've come from mathematics and I do physics and I do biology, and you know, really to master all these different disciplines is very hard. So I was very impressed already. So anyway, Ivy is a postdoctoral fellow at the Department of Integrative Biology and Physiology at the University of California, Los Angeles. She previously did a postdoc at the Department of Stem Cell Biology and Regenerative Medicine at the University of Southern California. She did her PhD at Oxford and her bachelor's in biochemistry at the University of Hong Kong. As I said, her work is super versatile, and she has used it to understand dynamic processes in biology and physiology. She has won several fellowships, the latest of which have been the Ludwig Cancer Research Graduate Fellowship and the UCLA Collaboratory Fellowship. And she has also been the recipient of several awards since early on in her career, and the latest includes the American Physiological Society Renal Section Research Recognition Award. She's also a lot of fun and very nice person, so I'm very pleased to introduce her talk. Yeah.
Thank you so much, Mariana, and it's my great pleasure to be here, kind of sharing my story with y'all. So I'm totally new to bioimaging, so the workshop was a very important moment for me to kind of launch my leap into imaging altogether. So thanks to BINA support, I was able to kind of, get into the field fairly fast, and has a very swift launch into microscopy. And ever since, like last November when we were in Irvine together, I was able actually to do some of the imaging myself, and also start to think about how we can combine imaging with mathematical modeling to more kind of delve into the dynamic processes in physiology. So today I want to kind of share a short story about how I'm using advanced microscopy to study sex differences in the kidney. So in the background is a light sheet scanning microscopy image of a cross-section of a neonatal mouse kidney. And we're staining for different vasculatures as well as the renal tubules, just to give everybody a flavor about the organ we're dealing with today. So a very brief background about kidney anatomy and physiology. So the kidney is a really interesting organ, and they are kind of characterized by these layered outlines. We call it different zones, especially the cortex, which is where all the filtration and reabsorption happens. So if we kind of delve into the anatomy, we usually kind of encounter these renal tubule structures, which we call nephron, which is highlighted in yellow here, where we have characteristic kind of very dense vasculature wrapped around it. So the nephron, or the renal tubule, is very important for handling the filtration of blood, which comes in through the renal artery, branching into this very dense cluster of capillaries. We call it glomerulus. So that's where the filtration actually happens. So the filtrate would go through this renal tubule in yellow, and a lot of the water and essential electrolyte will be reabsorbed, including all the sodium, glucose, etc. And one important kind of function of the kidney, of course, is to dispose the waste that the body generates, and also regulating osmolality, including blood volume and then composition, different electrolyte composition. And there are different dynamical processes that is important for this function, including filtration of plasma inside the glomerulus and mechanical sensing of tubular fluid flow. So as the fluid flows through this tubule, there are a lot of changes in terms of fluid flow, and the mechanical sensing of this fluid flow rate will be important for how to coordinate epithelial function for reabsorption. And of course, we have to reabsorb all the essential molecules, including water, salt, and glucose. And that's a very energy demanding process. So one of the key measures of renal function is called the glomerular filtration rate. Basically, we want to kind of capture how fast the filtration is happening and how much volume is entering the renal tubule at a given time. So we call it GFR, and that's one of the key indicators of renal function in the clinic. So one part I want to zoom in is this renal epithelial cells called the proximal tubule. So that's the proximal end of the renal tubule to the glomerulus. And we are kind of very impressed to see how structured those epithelial layers are. So they're characterized by this brush border anatomy. So it's an array of microvilli supported by actin bundles. So those microvilli together with the prima cilia, which is sticking out into the lumen, will be important for sensing the fluid flow and regulating ion channel activity on the membrane to regulate reabsorption activities through the apical side of the membrane. And if we look at the cross-section of those one epithelial layer, is densely packed with mitochondria to support this highly energetic process of reabsorption of sodium and the other essential molecules. And then one thing that we are very surprised to see the clinic is that there are very marketed sexual dimorphism in terms of disease profiles. So men compared to women have a faster decline in renal function that is age-related, and they also show high mortality rate and all levels of pre-dialysis chronic kidney disease. They show worse disease progression to end-stage kidney disease that requires dialysis. And there are also evidence showing there's an increased incidence of hospital-associated acute kidney injury, including like patients ICU going through ischemia-reperfusion injury. So the kind of key motivation of my research is to understand the sexual dimorphism in terms of renal disease profiles and trying to understand at the molecular level, the physiological level, why that's the case. So let's dive a little bit deeper into the cellular mechanism. So I'm very interested in this population of epithelial cells called proximal tubule cells. And they are very interesting in terms of how they organize their metabolism to support this highly energetic process of reabsorption. So they're packed with mitochondria, and they're kind of clustered towards the basolateral membrane, where there is abundant sodium pump, which is sodium, potassium, ATPase. So which requires ATP for pumping sodium across the basolateral membrane to create a sodium gradient, so that sodium in the lumen can flow through the apical membrane in a passive fashion. And together with that, we also taking in a lot of glucose, potassium, calcium, etc. And then another thing that is very essential for those epithelial layers is that they don't have glycolysis. Instead, they rely on lactate, which is freely available in the waste or the filtrate of glomerulus, to feed into the TCA cycle as a fuel for energy supply. So we have done a single cell multiomic study to look into male and female differences. And then one kind of key biological pathway we identified was how the male proximal tubule tends to use peroxisome lipid metabolism as their primary source of energy supply. But in contrast, female tends to actually get rid of lipid, and then they're trying to use lipid altogether. So that offers a hypothesis that male proximal tubule tends to utilize lipid as their primary source of energy, and is also densely packed with lipid droplet. So peroxisome is a specialized organelle that oxidizes long-chain fatty acids. So throughout this beta oxidation process, one key side effect is generation reactive oxygen species. So one of our hypotheses is kind of look into how this can actually underline the male bias renal vulnerability during aging. So this work kind of inspired us to ask, the following key research question. So we have seen all those different molecular differences between male and female kidney, but it's kind of unclear how those molecular differences would affect renal physiology, which is kind of a key scale, bridging the molecular details to organ physiology in the kidney. So that's kind of the question I'm tackling in my current research. So in terms of kidney physiology, one of the key lessons that we learn in all the kind of medical school or different textbook is how the kidney is very good at auto-regulating its blood flow. So one of those key processes is called a tubulo-glomerular feedback system, which trying to maintain fluid flow into these nephrons at a steady or within a physiological range. So there are different anatomic bases to this feedback. And one of the key interaction is this renal tubule. They go deep into the renal medulla, but they would actually loop back onto the cortex. And they are situated right adjacent to the afferent artery. That's where the blood comes into the glomerulus. So this little patch of cells called macula densa is very important for sensing sodium, and they will actually feed back onto the afferent arteriole to control the contractility of the blood vessels. So if you look through the micrograph of this little anatomic region where we have a patch around like 20 to 25 cells, they are the macula densa cells. They are very important for sensing sodium that is looping or is passing through in the distal tubule where the lumen is. So as sodium is rushing by, sodium sensing happens inside the macula densa, and they would send out signals to the adjacent afferent arteriole. And here's where the renin granule is. So they will control the release of renin, which catalyze the generation of angiotensin II to control the blood pressure contractility. So the whole kind of anatomic basis provide this very important negative feedback mechanism to control blood pressure, to control blood flow inside the glomerulus. So here's a simple diagram to show how that works. For example, we have an increased flow or increased GFR coming into the glomerulus. That will increase the tubular flow rate, and that will decrease the residence time of the fluid inside the lumen. Therefore, a smaller amount of sodium will be reabsorbed, leading to an increased concentration of sodium at macula densa, which is towards the exit of the tubule. So this high concentration of sodium will issue constriction signals to afferent arteriole, therefore decrease its incoming blood flow, which is decreasing the GFR. On the other hand, if we have a decreased GFR, the fluid flow will be decreased and increase the residence time of fluid in the lumen, therefore increase the reabsorption, leading to a decreased sodium concentration that is reaching macula densa site, therefore issuing relaxation signals onto the afferent arteriole. So this is kind of a characteristic negative feedback induced homeostatic control in the kidney, and many medical students are taught how this is important for maintaining the homeostasis in the kidney. However, it has been 40 years, and we are kind of start to see more and more clearly that the negative feedback can also generate robust oscillatory dynamics in the renal tubule. So here we're seeing fluid flow that is right next to or right at the exit of the Bowman's capsule going into the proximal tubule of the renal of the nephron. So if we have a sensor of fluid flow at this junction, we can actually see how this fluid flow actually going through this up and down patterns in fluctuation. And they have characteristic period around 45 seconds in terms of their oscillatory frequency. And even at a whole kidney level, we can also see traces of its evidence. So one of the popular physiological measurement in the labs nowadays for GFR, of a whole kidney GFR, is to have a transdermal biosensor, a sensor of fluorescence. So a single bolus fluorescence dye through the tail vein injection can be captured through this sensor on the back of the mice. So this is a typical clearance profiles where we see initial injection increase the fluorescence intensity inside the kidney compartment, and over time it will be cleared by filtration and then excretion into the bladder. So as the fluorescence intensity decay in the kidney, we can see that decay is far from smooth. And typically this is discarded as noise. However, if we mark this individual time steps in red, we can see that its zigzag also shows very robust oscillatory pattern. And they coincide with this period we're seeing on the left, around 50 seconds to a minute. So one interesting aspect about this GFR oscillation is that it seems to be essential for kidney function. And one of the early studies back in the 80s looking at this oscillatory patterns between normal rats and spontaneous hypertensive rats. And we can start to see that the hypertensive rats seems to have lost this regular oscillatory pattern. Instead, they gains a higher, different frequency spectrums and then show more kind of irregularity. And we can also plot out its tracings on a face portrait, and we can appreciate that instead of having this normotensive regular cycles, now we start to get a bit into irregularity and even in chaotic regime. So we hypothesize that one of the possibility that is leading to the male biased vulnerability in the kidney is that the male kidney, due to a higher energetic demand, they might be situating closer to the breaking line, which is demarcating the loss of oscillatory control inside the nephron. And the female resilience is conferred due to kind of more conservative energetic profiles, and they're situated further away from the breaking line. And this is a hypothesis we're testing very actively in the lab right now. And I would like to share some of the findings, some of the experimental setup we have in the lab to test this hypothesis. So the method I'm applying is combining intravital microscopy to look through, to look under the microscope how the kidney is undergoing filtration, reabsorption in real time. So a typical image on the left is kind of showing how we can actually look through the microscope to capture fluid flow inside the renal cortex. So different fluorescent dye can highlight different fluid compartment, including the capillaries, blood plasma flow in the capillaries, fluid flow inside the proximal tubule, or fluid flow that is in the distal part of the nephron. So the dynamic profile can be used to inform mathematical modeling, where we have a very kind of special team here to have a mechanism model of fluid flow inside the renal tubule, and then looking at how different parameters, especially the sensitivity of the negative feedback and how metabolic regulation would affect its dynamic property. So by looking at the oscillatory capacity, we can start kind of looking into how the male is more vulnerable in terms of age-related decline in renal function, as well as other disease profiles. So this has been applied before. So this is early image in looking into a glomerulus inside a rat kidney. So this is a glomerulus like situated the cortex. It's very superficial on the organ. And then what we can do is to track fluid flow inside different compartments. For example, we have fluid flow inside the glomerulus. We have fluid flow inside the proximal tubules at the bottom, and then we can also track fluid flow inside the distal tubule that's showing higher red intensity here. So if you aligned the different tracings, we can start to see that there are, we can capture the oscillatory fluid flow very well. And they seems to have a slight time delay, which is a physiological in terms of how the fluid is traveling along the single tubule. And more recently, there is a study looking at fluid flow on the microscope in vivo setting to look at how angiotensin II would trigger calcium activation inside glomerulus and how inhibition of angiotensin II receptor would abolish this calcium activation. So this is super exciting to see how intravital imaging can be applied to study a wide array of biological questions, and especially how the technology also matures over time to have more kind of in-depth with different genetic mouse lines. We can do a lot more as time goes on. So the setup I have is looking at a mouse kidney under the microscope. So I started with a very simple setup, just wanted to kind of see fluid flow, how it's being mediated inside the kidney during normal, kind of baseline condition. So here we have, on the left is kind of example of how we set it up. We have a mouse of different age, male and female, and we put them on the microscope. And here we have a setup from the earlier study, but it's very similar kind of across different labs. So the fluid flow on the right is kind of a typical kinetic video we can capture. So the region highlighted in white circle here, where we can actually capture multiple peaks in terms of fluorescence intensity, which corresponds to potential oscillatory control in terms of tubular fluid. And I have gathered longer videos comparing male and female, how they're actually processing fluid inside their proximal tubule compartment. And we can start to see that the male and female actually showing quite different dynamic properties in terms of how they handle their fluid. So here we can start to see how female seems to have a more robust oscillatory response, especially in terms of the amplitude of relative change in fluorescence intensity, and also slightly faster in terms of their frequency. So now we're doing kind of more in-depth analysis to compare and also kind of feed that into mathematical modeling, which I wish to share at a different time to all of you. Another aspect we're very interested in is to look into the metabolic basis of renal reabsorption, like solid transport activities. So we use the very special mouse line that has dendro-2, which is a fluoroprotein located into the mitochondria. So we can visualize individual mitochondria either in a snapshot setting, where we can have a more in-depth anatomic information, or we can use that for dynamic imaging as well. So here we are showing a cross-section of a very elegant glomerulus, where we see this kind of a lumen showing capillaries inside the glomerulus, surrounded by an array of renal tubules, including proximal tubule, distal tubule, as well as macula densa cells that we have seen earlier next to the afferent arteriole, characterized by kind of a endothelial marker. And we also stained it for a tubulin 3, which is nerve endings, to see like how heavily innervated the different anatomic site in the kidneys are. So here we kind of have an overview of the distribution of mitochondria, but we can also dive a bit deeper into individual tubules and then see how the mitochondria are organized among themselves, and how they are also interacting with each other. So on the right, we have a 3D rendering of a proximal tubule where we have multiple epithelial cells lined up facing the lumen. This is the apical site. And then on the peripheral, we have the basolateral membrane. And we have a hollow region where the nuclei are. We didn't stain it for, but we can kind of recapitulate where they are situated. And we can, I hope all of us can start to appreciate how densely packed the proximal tubules are in terms of mitochondrial content. And they show a very distinct morphology, which is intriguing in terms of how they regulate their metabolic activities dynamically. So one of the key interesting questions we're testing is how mitochondrial morphology are related to metabolic state. So I guess this audience has seen multiple images showing the filamentous mitochondrial network, especially in cardiomyocyte, skeletal muscles, and even in fibroblast. So they have a characteristic dynamic regulation among themselves, which we typically call the mitochondrial dynamics, in terms of their fission and fusion event to generate new mitochondria, which will fuse to facilitate different metabolic functions, especially that the elongated one, the fused version of mitochondria, tends to be more efficient in OSPHOS, but the fragmented mitochondria tends to rely on, like the cells showing fragmented mitochondria would rely on glycolysis. And there are in the kidney, there are marketed sex differences in terms of mitochondria morphology, and that has been done through cryo-EM studies. And also we are also starting to see how this mitochondrial dynamics are regulated circadianly. So in terms of our sex difference story, there are different evidence showing that sex hormones can actually regulate the fission process through activating MYC, especially the androgen receptor, which is very important in regulating renal sexual dimorphism. And another interesting aspect is how ischemia or oxidative stress can also trigger fission event. Potentially constant flow in terms of GFR can also trigger that due to mediating different metabolic demand. On the other hand, calorie restriction in the long term can actually promote fusion events to facilitate more efficient metabolic activities. And there are different evidence showing that in the kidney, if we have a prolonged fragmented mitochondrial state, that is associated with the proximal tubule atrophy, fibrosis, and chronic kidney disease. And that's something we're very interested to test in our system as well. So besides looking at how mitochondrial morphology is related to these metabolic activities, we are also very interested in looking into metabolic oscillations. And here are examples from different organ systems to show that inside a stretch or a strip of mitochondria myocyte, inside individual mitochondria, we can actually track how they are controlling their membrane potentials. So this will be TMRE capturing the inner membrane potential of the mitochondria, and DCF can capture ROS production together with NADH activities. So I guess everybody will start to see how dynamic those metabolites are, and their regulate seems to be in tangent in phase, or in another way to say they are showing synchronized activities. And this seems to be very important for cardiomyocyte function, especially during perturbed situation. For example, there are a shortage of blood supply, for example. On the right-hand side, we are actually having a very elegant example showing how mitochondrial activities are actually coordinated across different cells in the same epithelial layer. So here, the researchers looking at the salivary gland and how the salivary epithelials in the acini are actually synchronized in terms of their NADH and the delta psi dynamic profiles. And if we block the gap junction, which is important for their communication, we can disrupt their synchronization activities, which will be detrimental for secreting different proteins into the saliva. So towards the end, I want to kind of showcase how we can also apply different techniques to target different questions or more kind of demanding questions in the kidney. So here we have a study came out in 2017 where one group applied intramitral phlegm to look at how we can actually identify different metabolic signatures. So the metabolites, they show all the fluorescence, but the typical fluorescence intensity will be really hard to distinguish them because they have overlapping emission spectrum profiles. So the research proposed to look at the fluorescence lifetime and how different metabolites actually showing different decay profiles. And with this phasor plot, we can start to actually distinguish them. And here is an example showing the renal epithelial, different renal tubules can be marked, can be, kind of label-free, but identified through the phlegm approach to see how they are actually showing different metabolic profiles and also indicative of their activities. For example, the segment 1 and then segment 2 are actually in the proximal tubule actually have very different metabolic profiles in terms of their NADH, FAD intensities. And then distal tubule seems to be very similar to S1 segment, which is quite refreshing to the renal community. And here is in the same study where they use the same gating setting, the same gated setting for phlegm to identify different luminal fluid component, as well as different immune populations. And we can actually visualize over time how they are interacting in real time inside intact tissue in vivo. So that is very exciting to me and many other kind of kidney researchers, because that offers a very powerful tool to study dynamical processes in vivo. And of course, I'm sure this opens doors to study other organ systems as well. And I hope, I can kind of continue on this journey to apply a lot of the techniques I learned at a workshop, and also acquire new techniques along the way to facilitate my own research looking into the sex difference story inside the kidney. So with this, I like to thank my mentors. So it's a kind of a combination of theoretical mentors, mathematical mentors at UCLA. So that's Eric Dees and Alan Garfinkel. And then our imaging wizard is Natalie Perez-Leon, based at NIH National Cancer Institute. So she helped me to kind of set up the intravital imaging for the kidney, where she typically look at the intravital imaging in the liver, but it's really nice to kind of try the technique on different organ systems. And Alicia at the USC, she has been the kidney mentor for me over the years. So with this, I'd like to thank you for your attention, and I'm happy to take any questions.
Lovely presentations from both of you. Thank you.
Yeah, I thought you had an excellent talk. I had a question about the mice that were expressing Dendra2.
Right.
So one of the advantages with Dendra2 is that it's a photoconvertible probe upon excitation. So I feel like that would be a really excellent tool to do live imaging. Have you had a chance to use those properties of that fluorophore?
So I'm only kind of getting started. So the first stage is more kind of, take a renal section and look at their anatomic distributions. But the lab has already tried. So Natalie Perez-Leon, she has actually used that photoactivation or photoconvertible property of Dendra2 to study the diffusion or the fusion process of mitochondria. So if we activate it with a UV, like a specific mitochondria, and then we tracked it over time, so they will convert it into red. So the red signal would diffuse to other green mitochondria if they are fused together. So they have tracked that in liver, a salivary gland, but usually takes hours because it's slower in vivo. But I've seen examples, people applying that photoactivation process. And it's a very powerful tool.
Yeah, I asked because I actually use Dendra2 in my own research, but it's always in live cell imaging. I did not know that it's been expressed in mice, so I think that's super amazing and super cool. So yeah, thank you so much.
Thanks.
I just wanted to say I need to run to another meeting, but I'm very, very grateful. I just wrote it on the chat now. I will leave you for the questions. If you have any questions for me, I'm happy to receive them by email. But I just wanted to again emphasize my deepest thanks to Michelle, first and foremost, to Michelle Dickman, and to BINA for the support. And I'm sorry, I have to leave.
No worries, no worries. If anybody has time to stay on for questions, we have a little time too. Actually, Ivy, I had a question. Just obviously, this is a very complex thing, right? Because there's this intimate sort of connection between not just the kidney itself and blood pressure, but the kidney and presumably bladder pressure too. And one of the things that happens in males as they age versus females is that actually you start to build up pressure due to benign prostatic hypertrophy. And I'm wondering whether there's a downstream effect that could be also confounding this sex difference, because, not only are you potentially getting what you're seeing happening just basically from a metabolic standpoint, but you're also potentially getting some sort of feedback from the bladder that's saying, the bladder is full, it's not able to release, and potentially even the epithelia of the bladder are sending signals about the content in the bladder. So would it be feasible, because I know there are models for BPH, mice models for BPH, to see if you could actually exaggerate the sex difference using a mouse model that has BPH versus, comparing it to maybe age-matched female mice to see, again, if there's an additional compounding effect, because certainly in humans, I think that there's a lot of other potential contributing factors to that big difference between men and women.
Yeah, that's a very interesting thought. I would love to look into it. Of course, right now we're looking at the young in a more kind of, free ad-libitum conditions. So I wouldn't think that would be a kind of a determining factor. But as the animal age, if we want to kind of look at specific conditions, those mouse model would be having tremendous impact on what we can look at, and how the organ communication also important.
Yeah, it's beautiful. I mean, really lovely to see the quite striking differences at the cellular level. But as I say, that can even then be confounded even further as with the aging piece of it that you'll eventually take into consideration. Because as I say, I think that it is something that with age is much more prevalent in males than in females, as you say, that there's a deterioration in the kidney function, and kidneys are one of those organs that, again, once it's gone, it's gone. There's really very little recovering that.
We do have a quick question from the chat. Robert asked, are there sexual dimorphisms in the podocyte cells of the kidney?
Thanks a lot for the question, Robert. Yes. So we are not particularly looking into podocyte sex differences, but there are several labs here at the UCLA. They're looking into the role of estrogen receptor and how they mediate the podocyte functions. So you can check out Andrea Havanah's work together with Karen Roy at UCLA. So they are specifically tackling that question.
Thanks.
Very impressive imaging approaches to this, Ivy, really. And I'm delighted that, attending Michelle's course was able to help you with this. It's great work.
I only started since November.
That's amazing, really.
Yeah, Michelle has been really helpful.
Thank you so much, Ivy. It's fantastic work. We look forward to continuing our work together